Hey, I'm Erin Weisbart. I am the lead image analyst and a senior staff scientist in the Simony lab in the imaging platform at the Broad Institute of Harvard and MIT. Today, I am going to talk you through the JUMP image analysis pipeline and just sort of show you what it looks like and what's happening to the images as they go through this pipeline. So, as a reminder, this is where the pipelines are available in GitHub, the Broad Institute / imaging platform pipelines here in this Jump production folder. These are the pipelines that were used to make the Jump production data. So I'm going to show you now this jump analysis pipeline. So I have CellProfiler open and I have downloaded some files from the jump data set to my laptop. I have downloaded the load data CSV um, for these images. And what I have done is edited the paths in the CSV to point to where the images are on my local computer so that CellProfiler knows how to find them. And the load data CSV, what they do is associate some metadata here, batch, plate, well, site, um, are and source are the metadata that sort of define a unique image set. And then it lists the file names, and then the what we call it when it gets imported into CellProfiler, as well as the path names for each of those images. So, for example, the Illum AGP image is found in my desktop, this plate folder, and the file name itself. This is the name of the file name itself. And so I have, that's what is in here in this load data that I have told CellProfiler, load this load data so you know where to find my images. So I'm going to start test mode. Oh, looks like I have a typo. Didn't spell Brightfield correctly. So if we go, let's see, Brightfield. There we go. All right. So just if I change the name of the file, it clears the cache, and then I point it back to the file again, and now we should be able to start test mode. So it has loaded in the images of this first image set, and it is ready to run through the pipeline. So I have turned on all of the um, all of these eyeballs so that you can see what's happening in each step. And we're just going to step our way through. So if I uh, okay, another mistake. If we look in my, I just had the files in the wrong folder. So let's try that one more time. Um, so there we go. Okay. So it has loaded in the first. So this is telling me like load data is the first module. It's working right now on image set one, this group one, and this is the names file names that it has loaded in. This is metadata it's reading about the images, their height, hash associated with them, metadata that it brought in from the load data CSV, um, scaling information. So this is all automatically parsed and read by CellProfiler. Then in the next step, this correct illumination apply. So kind of hard to see with this. Um, Hopefully you can see what's happening here. So it has in this first column all of the images that it has loaded in as the raw images. Then in this middle column is illumination correction images. So these are actually NumPy arrays. That are loaded in that we calculate that get rid of any vignetting or any sort of debris in the illumination path in the microscope to make the illumination totally flat across the image instead of being, as you see, typically brighter in the center, darker in the outside. So these are very small values. You can see down here at the bottom, it's going to show numbers. And so as I hover here, at the very, very bottom there, you see the X location, Y location changing, and then the intensity. So all the way out where it's at the darkest, the intensity is 1. Up to an intensity of just over 2. So that means the middle of this image is actually twice as bright as the outside of the image. And so when we're making measurements on cells across the image, we want them to be, we want to help normalize that and not introduce artifacts based on where the cells are within an individual image. And so we apply this flat field correction. We take the input image, we divide it by the illumination correction image and put this output image. And then these images are what are moving on to the next module in the pipeline. So you can see what we set here in the pipeline for each of these is the input image, the name that we're going to give it after we apply the illumination correction, the name of the illumination correction function, and how we are doing the illumination correction. So in this case, we're dividing. So then now we are going to go to the next step, which is a measure image quality. So we have a couple image quality modules that happen here, and you can see what we set here is we can select the images that we are measuring. So we're measuring actually the input images before we apply any correction. And we have several different measurements that we have set here. And what one can do is use this information sort of in pipeline, or one can use this information downstream potentially to filter your downstream results. And so either to add on to a profile that perhaps there is information in the image at large, or to use as a sort of like quality filtering. So we think of these as sort of QC metrics. Focus scores tend to be, if the image is in focus, we expect to see, we can make some expectations about about our images to tell us whether they may or may not pass QC. And without going into that, that's a whole other talk. But you can use, intensity metrics if one channel has lots of saturated pixels, or perhaps it was a bad acquisition, or perhaps there was debris that is just getting in the way, and you can sort of use these metrics to check for those. And so if you have rules that you've set that so this is if you want more information about this, look up some information about like Cell Profiler Analyst is a tool that we developed that interfaces to help make QC rules based on image metrics. And so this would be a module that you can set rules for it to look and say, okay, based on these rules you've imported, this image that I'm currently processing probably is saturated, and it will throw a flag. Um, and then we have the option of either keeping the image in, but just adding an extra column um, in our analysis output data saying, okay, this hit this flag, I flagged this. Or you can actually tell it to skip this image if it fails QC. We don't have this module turned on in this pipeline because we haven't actually set all these rules, but just know that that's why this is here if you wanted to add that into the pipeline, but it's not currently turned on or running. So we then are moving into sort of uh, now a couple modules about identifying nuclei within the cell. So this correct, illuminate, calculate, this is different than a flat field correction. What this is sort of uh, basically you can see it sort of pixelating and then blurring out the image to help reduce or sort of help make structures more crisp, basically. So a good general principle in image analysis is you can do anything you want to an image to help you be able to identify objects in that image. So that's what this is for, to help us be able to identify nuclei within an image. But then when you make actual measurements after you've identified objects, you always want to go back to that original image, not the one you have modified for object identification. So this is we're getting ready to modify an image for object identification. So we input the DNA image, we made this sort of Illum image, and then in this next step, when we apply it, you can see that what it does, hopefully you can see a little bit, it takes this original image, we're just subtracting this little bit, a background subtraction of this illumination correction image, and then this image is just a little bit crisper for helping us find the nuclei edges. So then when we go into identify primary objects, we see that here we have input this corrected image. It is here circling all of the nuclei that it has identified. You can see here showing the objects colored, so you can see the different objects. And this is the nuclei that it has identified. So, so yay, we've identified nuclei in our image, and we can see that it does a good job with this data. If you have your own data set that you are wanting to use this pipeline on, this would be a module that you very likely would need to adjust. So object identification, you always want to make adjustments, tune it, do assay development for your data set. And so this would be a place where you would be adjusting pixel sizes, adjusting upper and lower bounds, smoothing, sort of all of these kind of advanced options you would need to adjust on your own data if you didn't see good object identification. One thing I do want to point out is that we have selected no, do not discard objects touching the border of the image. So all of the nuclei that we've identified here are currently kept, but bear that in mind because we're not actually going to make the measurements on all of these nuclei. But I'll walk you through there, but that's what we're starting with. We have kept in our nuclei pool here, these nuclei, including edges, includes nuclei that are actually partially cut off by the edge of the image. So then in secondary objects, we take those nuclei as seeds, and in a one-to-one relationship for every nucleus, we find a cell edge. So I'm going to adjust this contrast here, kind of blow it out so it's a bit easier to see. You can see this is what we have identified as the edge of the cells. We can zoom in and take a look and see that, if we adjust this contrast even further, yeah, that looks good, that we are getting, all the way to that, like what is actually a very dim edge of this cell. So this is good, accurate object identification. Again, with your own data set, this is something you would need to tune. And so this is cells including edges because it includes cells identified from the nuclei touching the edges as well. We then are going to filter objects. So what this does is filters the nuclei um, and then also applies that same filter to the cells. So we start with the nuclei including edges and we filter it to nuclei um, so that we no longer have the nuclei touching the edges in this in our actual nuclei objects that we're going to be using for object measurements. And you can see that, here's a cell that we have filtered out. It is not here in our filtered set. We started with 73 nuclei. Now we have 66. So that's what's happening there. Tertiary objects are basically, object math. So we take we define the cytoplasm basically as the cells minus the nuclei. So this is now a tertiary object that we have. So we have now cytoplasm to make measurements in. Then we are going to measure colocalization. So we have set measure all of these channels within all of these objects, measure here are all metrics. So what this outputs is a summary. So this is actually measuring on a per object basis. So we do have single cells, single cytoplasm, single nuclei measurements, but it summarizes them when we're in this test mode. So this is not what is actually going to be output to your spreadsheet or database in the end. This is a summary provided as we are going through. You can see, the measurements that it's making. We then measure granularity. Again, similar idea. Granularity has, is tunable. So basically all of these measurements are all tunable. And so it is with any image analysis, any morphological profiling you're doing, certainly elements made with the same pipeline, you can directly compare, between cells, within a batch, within a plate, but a different pipeline with different, both different image resolution or different, here things are subsampled and they have ranges, the range of the spectrum you're measuring in, or sort of a structuring element is part of the granularity measurements. So you may not be able to compare directly a granularity measurement from data set to data set. So again, this is a summary for each image, each of these granularity measurements. And when we export to spreadsheet or database, we will see it on a per object basis. Object intensity, same idea. This is not not as tunable, but we see these are all of the features that are created. So we get integrated intensity, mean intensity, standard intensity, min, max, intensity edges, quartiles, all in each of our objects, all sorts of measurements of different ways of quantifying intensity. Neighbors, here we are making measurements of neighbors counting cells, touching cells within a distance of 5. And so you can see this, we get here cells colored by the number of neighbors they have that are touching them, as well as cells colored by the percent of the cell body that is touching a neighboring cell. And so one of the, I mean, this is a helpful metric in morphological profiling because cell density can actually be a major confounding factor, as cells that are really packed in next to other cells can't grow to their full morphological potential if they're constrained in space. And so doing a neighbor analysis, or even as cell density analysis. This is often cell count in an image is often something that very much needs to be accounted for. Um, Likewise, we do this with nuclei. This can be helpful information for if there are help us understand over or under segmentation, help us understand if maybe a cell is actively dividing, or there are multinucleate cells that we're missing because of the way object identification we've done is, enforces a one-to-one relationship. But this is letting us know if a nucleus is touching another nucleus. Um, Finally, we um, uh, so this object neighbors, so this is cells within a specific distance, and this last object neighbors is, sorry, cells that are adjacent. So different ways of looking, again, quantifying neighborhood and adjacency. We then measure object intensity distributions. So saying, how is this intensity distributed throughout the object? We set a number of bins, and this tells us, so the innermost is the intensity, for example, here in the innermost part of the cytoplasm. Is it distributed evenly as you move from inside to outside of cell, or is it closer to the nucleus or closer to the edge of the cytoplasm? So you can tune the number of bins to tell you more information about where the object intensity is mostly distributed. Object size shape. This is, again, what you would think of, telling us information, stuff that's pretty intuitive, like the area of the cell, or the major axis length, minor axis length, down to sort of less intuitive things like Zernike's, which are shape descriptors. You get sort of complex math that can go on to describe shapes. And so often object size shape are described as being very interpretable features, which is makes them popular features. Lots of biologists, in particular, like having features be interpretable. And so there's often a lot of emphasis on size and shape features because of that, versus things like texture end up being quite a bit more abstract in the math that it's measuring. I guess as I'm mentioning it, I will point out that for each of these modules, so again, here is texture measurements made in these channels, both within the image and then within the objects. Um, then if you right click on any of these, you can always get to help and any module, and we have extensive documentation that tells you more about what any of these individual measurements are. So that's a helpful thing. Wanted to remind you of that, particularly for less intuitive features like texture features, can um, tell you more specifically what is the math that is going into this individual feature. And and honestly, I don't understand. You don't need to understand. You don't need to actually have them be interpretable necessarily to to have them be helpful by descriptors of biology. But this can tell you more about what's going on. You'll notice you can measure the texture in different gray levels and measure it at different scales. That you can really, read our documentation for more information about why you might want to tune these different parameters. Also, every parameter, see, has a little question mark next to it and will tell you information about that parameter in particular to really help you tune your pipeline and or make different measurements if you want to either understand what's here already or understand how you might want to modify it for your own purposes. So we have now also this sort of small chunk of features that are specific for measuring mitochondria. And what we have done here, we'll adjust this contrast so it's easier to see. So we apply this enhancer suppress that is enhancing tubeness. So it takes the signal in this mitostructure and applies a filter to it that makes it more tubular. We then threshold it. So this is this tubeness image input. It's thresholded into black and white. Then we create a morphological skeleton. So it takes this black and white image, and so we can zoom in here, actually skeletonizes it so that you can see this descriptor of the mitochondrial, the connections in the mitochondrial structures. And then we measure the object skeleton. So it's finding, this is showing you, we'll zoom in, showing you that it's finding branch points at the nucleus are these red dots, branch points in the skeleton trunks. Um, uh, yeah, are also red. And then branches have, oops, green endpoints. Um, branches are great. Oh, branches are green and endpoints are blue. Anyway, it is finding all of these different descriptors of this skeletonized structure to help describe the mitochondria in great detail. And then the final, like special mitochondria module is measuring intensity distribution within this tubeness, mitotubeness feature, which was this first one where we had just done an enhancement. And so again, if you may remember me saying in the beginning, you only ever want to make your measurements on your raw images, right? So here, this is actually an exception to that rule. So we have the mito channel that is treated like all the other channels in all of these other measurements where it's being made on the raw image. And then these five special modules here were actually developed to explore, specific aspects of mitochondrial biology. And so we are actually making a measurement on a modified image. So exception to the rule. But this is sort of a sub assay that was developed, for a specific target, a specific biological question. So that's what these special mito modules are. Um, then we apply a bunch of masks to our images, and this is because, so I'm gonna adjust the contrast so you can see it here. There we go. So we want to be able to make measurements on just the background of the image where we have masked out all of the cells. And so that's what we're doing here. This mask image, we're taking the DNA image. We are making DNA background only by using these cells, including edges, object masks, and applying those masks to this input image. So then when I, blow out the signal, what is left is just the background of this image. Likewise, we do that with AGP and bright field and the various bright fields that we have, the mito, the RNA channel. So all of these, it's here looking at bright field. Again, if we adjust the contrast, we can more easily see what's going on in the other channels. And so we have these background only images, and then we're measuring the image intensity across the raw images as well as this masked background only images to see if having information about the non-cell parts of an image actually help us in creating a profile or in running QC. Finally, we overlay some outlines onto blank images. So we have nuclei outlines, we have cell outlines, and we're overlaying these on black images so that they can be saved out with these two save image modules. So their output when we run the pipeline so that we have the actual objects that can then be used if somebody wants to import, import these outlines for their own, for a separate downstream image analysis task. And then finally, this export to spreadsheet module, this won't run when I'm in test mode, but what this does is saves out all of the measurements that I have made while running this pipeline. We have it set to export specific CSVs, which are just the ones we care about, the nuclei, the cytoplasm, the cell measurements, as well as information about the image and the experiment itself. We are not, so there are other data set options. We are not having it export information about these cells, including the edges, or the nuclei, including edges. We don't have those save out because that was just used for creating those masks. So we're not saving out data that we don't actually care about. Likewise, we have selected specific measurements that we want to save and export within here. And one can choose to just save out all or select measurements that were made that may be duplications. And the reason we do that is that often the data is compiled into SQLite files. And if there are more than 2,000 columns, it's challenging. And so by taking the output CSVs and putting them into the SQLite database, we are limited in the number of columns that we have and can export. We're working on standardizing workflows that doesn't have this limitation, but that's why that was added to this pipeline. So that, in a nutshell, is the JUMP cell painting analysis pipeline.